So today we're going to be beginning the second half of note 4.1 from our grade 11 biology course, where we're going to have a conversation about the structure of genetic material. So I want to start off first with a super important definition, which is genome. So you may have heard or read in the news about the Human Genome Project and wondered, well, what does that encompass? What is that about? Well, a genome, by definition, is the complete DNA sequence of an organism. So in other words, it's sort of like the map of the DNA for a particular species. So if we're talking about the Human Genome Project, that was a project that involved mapping out the entire DNA sequence for humans, which is pretty cool. And we've been able to access a lot of information about genetic disorders and genetic linked conditions through just identifying the human genome, which is pretty cool. All right. So another important term that we need to know, I mentioned DNA above, and we all talk about DNA as if everybody understands what that means, right? And so DNA is an acronym, and it's an acronym that stands for deoxyribonucleic acid. So the deoxy is the D, nucleic, and then acid. So that's what DNA stands for. And so students often struggle with, well, what is the relationship between DNA and chromosomes and chromatin? And so I'm going to explain that to you, hopefully, so that it's a little bit more clear. So we know that during interphase, our DNA is going to exist as thin strands of what? Well, what do we call those? When DNA is in its thin strands, we call that chromatin. So let me write that out for you. So really, if we're looking at the relationship between DNA, chromosomes, and chromatin, we're all kind of talking about the same thing. And I'm going to look at a diagram in a minute, and I'll help to kind of break those ideas down for you. Okay, so if I'm talking about a chromosome is simply what we get when we take our chromatin, which is simply those thin strands of DNA, and we supercoil them, as you would have seen in yesterday's video showing you the supercoiling of DNA. So what happens is to create that chromatin is that we take molecules of DNA. So DNA is the molecule that contains all of our genetic information. When we take DNA and we wind it around these proteins called histones, what we get chromatin. And then when chromatin, which is basically just thin strands of DNA wound around our histones, when we take that and we supercoil it, we end up with a chromosome. So really, we're all talking about genetic information, whether we're talking about the DNA molecule itself, the chromatin, or the chromosome. It's all just genetic information in different forms. Okay, so during mitosis, our DNA we know is going to become supercoiled, and then we're going to begin to call it a chromosome. So often I'm asked about, well, how big is this? How long is this? And so I've included a diagram for those of you that are interested to see some of what the measurements are. So you can see here that the diameter of a DNA molecule, which we're going to get into, is about 2 nanometers. So you'll remember from grade 9 and grade 10 science, we looked at units, that 1 nanometer is equal to 10 to the negative 9 meters. So we're looking at a really, really small measurement there, really small diameter. Moving on, we look at our histone fiber, and it's going to be 11 nanometers. So already we're wrapping things up here. Then we get to what's called a nucleosome. When our chromatin begins to start to coil, we're looking at 30 nanometers. Moving it down to a 300 nanometer coiled chromatin fiber. It begins to coil again here. We're seeing it increase in size, so that's becoming more and more visible at 700 nanometers. And then eventually, if we're looking at one of the sister chromatids in our chromosome, at metaphase, we're looking at about 1400 nanometers. So small, but quite a lot bigger than it was when we're looking simply at our DNA molecule on its own. So in a minute here, we're going to be looking at some descriptions of DNA and another diagram, but I thought I would start out by giving you a simpler diagram before we move into a more complex one. So DNA is essentially, as you saw before, a bit of a ladder that's kind of twisted. So I want us to envision that we have a ladder that is untwisted, kind of to start with. So a ladder has the two sides, and then it has the rungs or the steps in the ladder, right? So imagine that we've untwisted our DNA molecule, and it looks a bit like a ladder. Now what I want to do is I want to zoom in on that ladder to see what the component or constituent parts are of it. So if we were to zoom in on the parts of the ladder, right, what we would see is that the sides are made up of specific structural components. So what you will see on the sides of the ladder is you will see alternating phosphate, I'm going to put a P there, and then a sugar molecule, I'm going to put an S there for sugar, and then a phosphate, and then another sugar molecule. That's the basic structure of the sides of our ladder. So really, this right here is going to represent maybe one side of our DNA ladder. Then of course we'd have the other side mirroring that. So I'm going to try something I haven't done here before with this. So I'm going to see what I can do. Did it work? Ooh, it didn't work it wasn't too bad. Okay, now that would actually be upside down and backwards. Let me see if I can rotate this. I'm going to see if I can rotate it. Well, I don't seem to be able to do that, but that's okay. And so then what we see next, so I want you to, we'll see this in the real diagram, but here, I'm just going to move this a little closer. And then now what I want to do is I want to get into, well, what is making up the steps of this ladder? What types of molecules are involved there? Well, making up the steps of the ladder, we have what are called nitrogenous bases. So they're nitrogen-containing compounds, and there are different types of them. And they're actually connected molecularly to the sugar in those sides of the ladder. So, and we have different types. So some of them are wider or double ringed, and some of them are a little shorter or single ringed. And so we have 4 different types of these nitrogenous bases, each making up a different part of the step. There we go. So on and on and on. So when we're going to look at the specifics of this in a second. So for right now, what I want you to understand is that the sides of the ladder are made up of alternating phosphates and sugars. Those are the sides of the ladder. And that the steps in the ladder are made up of a molecule called a nitrogenous base. And there are 4 different types of them. All right, so in terms of DNA structure, let's review a few of the ideas that I just discussed in drawing my diagram. So first of all, DNA, when we discuss it and we discuss its shape, we typically refer to it as a double helix structure. So it's a helix, like a bit of a corkscrew, and it has 2 sides of it. So it's not just a single corkscrew, it's a double corkscrew. Okay, so 2 strands and then twisted together, joined by the nitrogenous bases. So DNA is made up of repeating units called nucleotides. So a nucleotide would be, if I were to just talk about a single phosphate attached to a single sugar attached to a single nitrogenous base. That would be what we would call a nucleotide. All of that together would be one nucleotide. And so when we talk about DNA structure, it has repeating units, each called nucleotides. So in other words, we would take a multitude of these individual units and have them repeating along the sides of the ladder to make up the entire DNA molecule. But a single nucleotide, as we talked about, is made up of a sugar molecule. In the case of DNA, that sugar specifically is deoxyribose. Okay, so when I'm talking about the sugar molecule, Here I'm talking specifically about deoxyribose. Now, next, we have a phosphate group. And there's our phosphate group up there. And lastly, we're going to have, as I mentioned, one of 4 nitrogenous bases. And so they have specific names, and I'm going to talk a little bit more about some rules around nitrogenous bases in a minute. But we have 4 of them: adenine, guanine, thymine, and cytosine. But for this year, it'll be sufficient for you to remember A, G, T, and C. And so those are the nitrogenous bases. So when we look at our DNA molecule in this diagram here, you can see that, there are 4 different colors: green, red, black, and blue. And each of those are meant to identify a different nitrogenous base. So in this diagram, for example, adenine is colored in green, thymine is colored in red, and so on and so forth. Okay? So there's a few things that I want you to notice here, which is that adenine is always in the same rung or always paired with thymine. Adenine and thymine are always together. By the same token, we always see guanine and cytosine paired with one another, right? So that's what we call base pair rules or base pairings. So there are some rules around that. So adenine will always pair with thymine. I just always remember it makes the word at, and that seems to help me. So at, and then G always pairs with C. And that's what we refer to as complementary based pairing. And we're not going to get into the details of why A only pairs with T or why C only pairs with G. That will come in grade 12 biology. So here's another picture of our DNA molecule where you can see the double helix structure. Here the structure's been unwound. And what you can see here is, here we have a nucleotide, which is that repeating subunit of DNA. But we can see that the sides of the latter have that sugar phosphate, and so on and so forth. Now, as I mentioned, and I wasn't able to do earlier, you can see here how the opposite side is sort of upside down relative to the first side. And that, again, is important for you to note and remember for grade 12 because it will be part of our discussion then. So, again, we can see the complementary base pairing here because what seems to be labeled as C here is always paired with the green, which must be G, right? One of these is our adenine and the other one is our thymine. And so on and so forth. I'm going to post the link to this video on the structure of DNA in case you need to visualize this a little bit better in an animation. You'll have a YouTube video to depict this for you so that you can, and that might help with your comprehension of these ideas. So at this point in the lesson, I want to talk about chromosome pairing. But first I want to recap on some ideas that we looked at in the last unit. And the first is a couple of key terms. So the first is haploid. And the second one is diploid. And so you'll remember from the last unit that haploid refers to a cell having half the number of the chromosomes for that organism. And diploid means having the full complement of chromosomes for that particular organism. So if we were to look at a cell for each of these, I'm going to draw a haploid cell and a diploid cell. So in a haploid cell, we would have half the number of chromosomes compared to the diploid cell. So for instance, if my organism only had 1 chromosome, I'm going to show it as double-stranded, then my diploid would have 2, would have basically 2 sets of chromosomes. Now, I want to remind you that haploid cells in humans or in other organisms are typically, those are our gametes or our sex cells, reproductive cells. Whereas diploid cells are just our regular body cells. So I'm going to write just like other than sex cells. So for instance, skin cells and so on, and muscle cells and so on and so forth, right? So in order to make you or any organism that is produced via sexual reproduction, there had to have been one sex cell from your mom and one sex cell from your dad. So for instance, this might have been dad, and this might have been mom, and then the two of them will come together to form the first cell that might be you, and then you go on and duplicate from that one cell. So it should make sense to you that in every diploid cell in your body, the set of chromosomes that you have two sets. Half of each set is a chromosome from your dad, and half of that set is a chromosome from your mom. Right? So when we talk about chromosome pairing, we're talking about the fact that you should have two sets of chromosomes, one from dad and one from mom. So it's important to note that, and you probably are aware of this, that the number of chromosomes is going to vary from species to species, and it doesn't reflect the complexity of the organism. I'm going to pull up a browser here to show you what I mean by that. So what I've pulled up here is a list of organisms by chromosome count, just so you can see. And so, some organisms have very few chromosomes. Like for instance here, we're looking at a different ant. But I'm going to scroll up. You can see as we go, this is by increasing number of chromosomes. So I'm trying to see if you remember, well, how many chromosomes does a human have? And I'm hoping that you remembered 46. So I'm just going to scroll to 46 here. Right, so here we are, humans, with 46 chromosomes. And then I look down a few rows and I see that a potato has 48. Well, I like to think that humans are far more complex than potatoes, capable of free will and all, and potatoes are not. So again, that's just one easy comparison to show you that the number of chromosomes doesn't always correlate to the complexity of the organism. I'm just going to see if I can scroll through any more here to see. There's a pineapple with 50. I wonder which organism on this list has the greatest number of chromosomes. Oops. It looks like it is the ciliated protozoa with 1,260 chromosomes. Well, there we go, ciliated protozoa. So kind of, I'm guessing, almost like a paramecium of sort. Right, here we go. So, there's just an example to give you some frame of reference around number of chromosomes and complexity and the fact they're not really related. Okay, so moving on with our lesson. As we said, we usually say that humans have 23 pairs of chromosomes rather than saying 46. Well, we can say 46, but really what we mean when we say 46 is that we have two pairs of 23. And remember, one set from mom and one set from dad, right? But one of those 23 sets are responsible for determining the sex of the offspring. So we know that if you were to line up all of your chromosomes, each of the 23 pairs, you would see that the 23rd pair is your sex chromosomes. So if you have an X and an X there, then you would be female. If you have an X and a Y, then you are male. So this is just I believe this is just under a microscope or some kind of really intense microscope. This would be a male set of chromosomes, because you can see here's our X, and then this is what the Y would look like. It's sort of like a truncated X of sorts. So we say that the other 22 pairs, aside from those that determine your sex, are called autosomes. So your autosomes are all of the other chromosomes that are responsible for a variety of other traits that are you, but they're not involved in determining whether you're male or female. Okay, and so we have autosomes, and then we have sex chromosomes, and that's it. So another thing that I wanted to mention was a karyotype. So you've probably seen a karyotype. I'm just going to move this up so that you can see it. This is in your textbook on page 167. And so let's take a look at this to see, well, is it a human karyotype? A karyotype is basically just a picture, a complete picture of a person's set of chromosomes, whether human or not. Right? So if it's a human, we're going to expect there to be 23 pairs. So here we can see the first pair, second pair, third pair, fourth, and so on, all the way down to the 22nd pair, and then the 23rd pair. In this case, this is showing an X and an X. So this karyotype as a whole would be that of a woman. If this last 23rd pair looked like this, then that would depict a male. Okay, so now there's some other things that I want you to notice here on this karyotype. How are the chromosomes organized? Well, what I'm hoping that you'll see, aside from the fact that we have our 23rd pair, which determines the sex, I'm hoping that you see that they're organized in order of largest to smallest. And also, they can be organized based on the position of the centromere. The centromere, of course, is what joins our sister chromatids together. So you'll notice that some chromosomes, though they are the same height roughly, will be organized based on the position of their centromeres. And that actually helps with pairing chromosomes as well. So a term that we use to describe those chromosomes that are paired up is the word homologous. We often refer to them as homologous pairs. And that is because they have the same type of genetic information contained on them. That's why they are paired together. So for instance, if I'm talking about, one particular set of chromosomes, let's just say this is chromosome pair number 1. Perhaps, and there's a way of looking this up. This is just strictly an example. I would have, one chromosome from your dad and one from your mom. But regardless of what, whether it's from your mom or your dad, and regardless of whether the information on each of these is the same, you would still find the same kind of information for a particular trait on homologous chromosomes. So for example, if I were to maybe zone in on this section of dad's chromosome, in the homologous pair, your mom would also have a similar gene on her chromosome that's paired, right? In that homologous pair, we're going to see the same type of gene. So for instance, maybe we're looking at what I've highlighted in green might be the sequence that will determine your eye color. So perhaps, but it doesn't mean that it's identical information. For instance, your dad might have a gene that codes for blue eyes, whereas your mom might have the genetic code that codes for, let's say, green eyes. And that's just one example. Okay, so what is a homologous pair? Those chromosomes are homologous because they are paired together and they contain similar genetic information. So some other points to take note of is, and this is a diagram to your textbook, so I'm going to try to break it down for you, is that homologous chromosomes, and of course here all we're looking at is a single strand, and what I drew was the same idea, it's just that these chromosomes have not yet duplicated. But what we would be looking at is we would pair chromosomes up and determine that they were homologous based on a few identifying factors, one of which is length, and another one of which is our banding pattern, as we saw in the karyotype. So these two chromosomes might be identified as being the homologous pair because of those, as well as the position of the centromere. Okay, now those bands may describe what's called an allele. An allele is a term that I'm going to define in a minute, but an allele is essentially just genetic information. Let's take a look at that now. So I've taken the liberty of drawing another pair of homologous chromosomes. Now, in this case, again, I'm just showing them as duplicated as opposed to the single strand. So each chromosome in the homologous pair carries a unique allele, and allele is just a different form of the same gene. So really, when I was talking about, maybe dad having the code for blue eyes or brown eyes, I can't remember what I said, and then mom carrying the code for green eyes, what I'm describing by saying the blue or the green eyes is just information relating to that particular allele. So an allele is basically just a code to an extent. Okay, so a homologous pair, one chromosome is going to contain the allele from your mother, and the other chromosome is going to contain the allele from your father. That's all we mean when we say allele. Okay, so the allele for brown eyes, oh, see, now I've switched it, so let's cross that out and say, instead, let's say green eyes might be for mom, and and dad might have the allele for blue eyes, and that's just one example. So I know that was a bit to take in. I'm going to suggest to you that that be it for today. Aside from some homework questions that are just going to simply probe your understanding of these ideas and ask a little bit more of your thinking skills around these ideas. But as always, reach out with any questions that you might have, and I'll be happy to get back to you as soon as I can.